Hello everyone. Welcome to my YouTube channel. In this video, today I'm going to uh, give you information about the microalgae protein production, uh, on which I will let you know about how chlorella could be uh, could feed the future. So, in this video, you will learn about fully Chlorella vulgaris omics. So, what if I told that a microscopic organism could produce protein more than a soy, or grow faster than crops, and that needs minimal land, even in a diverse unusual environmental condition? Then definitely we have to talk about the Chlorella vulgaris. This is a single cell microalga. It is becoming a major player in the future for the sustainable protein products. Okay, so today I'll let you know how we can produce protein and how scientists use omics technology to optimize, and how it could be transformed into food, feed, and biotech industry. So, Chlorella vulgaris is a freshwater microalga known for fast growth, high photosynthetic efficiency, and extremely rich in the nutritional composition. They contain almost 50 to 60% of protein by dry weight, and depending upon the cultivation. So that is comparable, or even higher than many plants. But it makes it is not just a quantity; it's a quality type. So it contains essential amino acids, bioactive peptides, and antioxidant. How protein is produced in chlorella. So to understand the basic mechanism, protein production in chlorella happens through the photosynthetic and metabolism. Okay, the first step for the protein production is carbon fixation, on which carbon dioxide is fixed into organic molecule inside chlorella vulgaris, and nitrogen source will be like nitrate and ammonium that convert into amino acids. And primary protein synthesis uh, occurs through the assemblage of amino acids when the amino acid became together. So to optimize, we need to have like a optimize the condition of nitrogen, optimize the nitrogen supply that can significantly increase uh, the boost of a protein production, because if we have a more nitrogen, then we could have a more amino acid that result in the protein. Studies show that the protein productivity increase 4.8 under optimized conditions. It means when we have optimized environment, there will be high possibility of increasing protein. So under conditions, protein content can be reached up to like 58% if we supplied enough nitrogen to the Chlorella vulgaris, even other necessary supplements or nutrients. Roles of omics technology. How omics technology will play a role for the optimization of protein in a Chlorella vulgaris. Like scientists don't grow the microalgae, they analyze the multiple levels, how it can be improved. Like genomics identify the genes that is responsible for the protein synthesis, which gene is responsible. Through that, they can modify or optimize or utilize such kinds of genes, and upregulate those genes through the upregulation of those genes that will enable to increase the protein synthesis. Transcriptomics will activate under high nitrogen or stress condition. What kinds of situation does Chlorella vulgaris or even a microalgae face? Like under nutrient stress conditions like nitrogen, there will be chances of increment of fatty acids. But in case of excess of nitrogen, or in under high nitrogen condition, that will case is one case for increasing carbohydrate boost off and protein productions as well. Proteomics measures the actual protein being produced by Chlorella vulgaris through these techniques using like gas chromatography, mass spectrometry, different chromatographic technique, and metabolomics plays a very important role to track the amino acids and the metabolic pathway which are activated or which are active state and playing role. Uh, so why does it matter, the omics technology? Because through the omics technology we can optimize the growth condition as well, and through the genetic engineering we can have like high protein producing stress, and we can understand the stress response as well. So there are so many ways to grow Chlorella vulgaris. We can grow Chlorella vulgaris in phototropic condition in a photobioreactor using light and carbon dioxide, and also in the heterotropic conditions in the absence of light uh, through the supplement of organic compound in the dark state, and as well as it is possible to grow Chlorella vulgaris in a mixed state as well, like through the mixotropic growth, supplying light as well as organic compound. So advanced strategy includes like currently developed photobioreactor system through which we can grow Chlorella vulgaris without problem. I have to say like that, if we could optimize the environment, unless optimized condition, there will be some of the challenges which the world is currently facing. In case if we grow microalgae on restway pond system, but this kinds of system is quite affordable, as well as for the optimization of growth of those kinds of microalgae, whether it is in a photobioreactor or in a reciprocal system. It is necessary to have like enough light intensity, carbon dioxide concentration, nitrogen level, and pH to maintain the optimal growth. On proper optimization of the environment, we can have the expected biomass as well as content of a protein that we have desired, and that could be become accessible as well. How can we extract those kinds of substances like proteins, carbohydrate, even lipids? For development of those kinds of compound from the microalgae, there is a huge challenge which we have to tackle. So for that, we have to destroy the rigid cell wall of a microalgae. It can be possible through the destruction techniques. For that, we have to implement a high throughput technology, as such as through mechanical bed milling, ultrasound, and enzymatic mm, degradation process. As well as we can go through the like extraction process using different kinds of enzymes and proteins that also can help to degrade or denature, even or destroy, break down the cell walls of microalgae, which is uh, difficult to destroy and extract a high valuable compound. So for the separation and purifications, same we can utilize the same new technologies. We can go through like heating. This is the advanced technology which we resolve the bioactive compound, as well as saves energy too. Beside that, the electric assisted other flotation techniques which could increase the protein content, through the recovery process. And we can use integrated system so that we can valorize the whole biomass of a microalgae. The Chlorella microalgae protein is mm, a highly demanded substance, even a valorized compound. Like they are responsible, and even they are very important in the industry like food, as a protein powder, meat alternative. In aquaculture as well, they can be used as a fish feed. Pharmaceutical, we can utilize as a bioactive compounds. In sustainability, we can produce more protein for more land area, and even we can have a 15x higher yield per hectare. So, despite the promise, challenges remains in microalgae cultivation. What are the challenges the microalgae industry at this time facing on, like a high production cost, complex downstream processing, scaling from lab to industry, and even a customer acceptance as well. So, to uh, enrich or increase the Chlorella vulgaris protein in future, what should I have to do? We have to go through the Omics technology and bioprocess optimization. This process might help us to improve the production of a protein from Chlorella vulgaris. Thank you so much, everyone, for your attention. I will be back with another new video with a new topic. Thank you so much again, and please hit like, share, and subscribe.